So our next speaker is Joel Voldman, Associate Professor of Electrical Engineering and Computer Science. He works on the intersection between biology and microtechnology. And I think we're going to hear about insights made in cells.
Oh, no, I'm good. Can I close it? You sure? We're still on the other one. That's our problem. How's that? All right, well, I appreciate the opportunity to talk to you for a few minutes. I actually don't work in cancer, but the reason that I'm here is because I was reading these NCI provocative questions, and one of them asks whether the physical properties of tumors, and specifically they mention electrical properties, could be useful somewhere. And that sparked my interest because that's one of the areas that I work in, and I want to tell you a little bit about that today. So physical properties, why would we ever look at them? Well, you just heard about phase contrast, or that's one physical property, size, shape, and I'm going to talk about electrical properties primarily. And there's three reasons that I could think that you might want to look at them. One is specificity. If the phenotype you actually care about is a physical phenotype, then you might want to directly measure that property. And so, for instance, people looking at differentiating CTCs based on size. Okay? Label-free, so you don't have to add a label in order to make the property apparent. And so if you want something where you want to do fast assay or minimal sample prep, that's a reason that you want to look at physical properties. And the third one is if you want to do something where you want to reintroduce the cells and you don't want to either do transgenics or add a label that's going to go back in, well, then again, physical properties are easy. Okay. And I'm going to talk about electrical properties. So everything in this room has electrical properties, vacuum, this lectern here, as well as cells. Cells, to first order, are membrane-covered spheres. They have a fairly insulating plasma membrane and then an internal cytoplasm. And each of those has a conductivity and a permittivity associated with them. And if something biological changes one of those properties, you'll see that in a change in the electrical properties. So for example, if the cell goes through apoptosis, punches holes in the membrane, you're going to see that as a change in the electrical properties. And that's not new, it's physics, it's been around for a long time. Okay? So how do you actually make those changes apparent? What's the technology like? And one approach, and the approach that we use, is this thing called dielectrophoresis, and I'm going to do a little bit of cartoon physics here, but if you have a cell, this green ball, and it is lower conductivity than the liquid that it's in, then the field lines will wrap around it. They don't want to go into the cell, and the cell will be pushed in one direction, in this case down. Okay? On the flip side, if the cell is more conductive, it'll be pulled up in this situation. Okay? Do that. This translational force that arises is actually called dielectrophoresis. It's been around, again, as well for about 40 years. It can be cell-friendly. It's good for manipulating particles that are of the size of cells. And you can tune it by varying the frequency that you apply. Okay? And the key for what we're going to do is that if the cell is actually in a liquid or medium that's the same as the cell, then it's transparent. Just like if it has the same index of refraction as the medium that it's in, you can't see it. Okay? And so what we can do is we can set up a situation where we have a gradient of electrical properties in a liquid. Okay? And we have a heterogeneous population of cells, and we can force them to migrate to where they're electrically transparent. And at that location, which will be independent of their size, you'll get a measure of their electrical properties, okay, or their polarizability is what we call it. This is analogous to what you'd see in isoelectric focusing, where you separate proteins by putting them in a pH gradient, applying an electric field, you separate on charge. Here you're separating cells based on their polarizability, their bulk electrical properties, okay? The method that results from this is called isodielectric separation, and the way it actually looks in practice, it's a little microfluidic chip. You have little electrodes in it. You have you set up a liquid conductivity gradient using very standard approaches. And when cells come in, there are different electrical properties. They will migrate to different locations in the channel, and they'll be continuously separated. Again, you can image that over time, and here's potentially a movie that somebody can see of two populations, and as you vary the frequency, the separation and the location of those populations changes. And so if you have a population, you can get essentially a histogram of the distribution of the properties on a per cell basis. Okay. So I'm going to give you 3 snapshots of how we use this that illustrate different aspects. One of them is a project we have on sepsis. This slide is a lot of stuff, none of which is important to remember, except that there's this project where we want to actually do therapy for sepsis, and as part of that we want to do sort of very fast measurements of the state of the patient. Okay? And that leads to one of those reasons that we want to use physical properties, because we don't have to label, so we can do things quickly. Okay? And so what we have done is looked at whether we can monitor activated immune cells using electrical properties. We have this mouse model of sepsis that we can induce sepsis in these mice, and then we can assay them in assay the WBCs in a number of different ways, and we compare flow cytometry to what we get from our little isodielectric separation device. We first have found that in humans, for instance, these are human samples, fresh blood, that if you activate them by hitting them over the head with PMA, you can actually get a difference in the location or the electrical properties of those cells that's repeatable and varies with frequency. And you can also get that with mice. So on the x-axis is what you get in terms of activation from flow. On the y-axis is what you get from our little thing. Flow takes about an hour, hour and a half to run, including the sample prep. We can do our assays in about 5 to 15 minutes. Okay? So you see that you get a variation depending on the duration of this CLP, this CLP model. And then if you look at the overall correlation, you're not doing too bad. It makes our DARPA people happy. Okay, so that's one snapshot, and the take-home message is you can actually use this, at least in a preclinical model. There may be some utility here. Okay. The second one is, well, can we, what's the smallest difference that actually results in an electrical difference? And so what we did is we looked at the yeast deletion library, and we looked for what single genes deletions can actually productively affect the properties of the cells. I won't show you the screening methodology, but it was fun. But anyway, we got a bunch of hits, okay? So we have about 400 genes out of the 4,500 that we got whose deletion significantly changes the electrical properties of the cells. I won't show you the validation that we did, reintroducing them, taking advantage of the yeast genetics. But the point is, here we have a list, okay? And there may be homologs in human, or you can imagine doing a pooled shRNA screen in human to look at, well, are there gene deletions or knockdowns that actually affect the electrical properties of cells that might be useful in a diagnostic or screening application. And the final thing I'll tell you about is that, well, you can also, just like in flow cytometry, you don't typically run one marker. Well, there's no reason to run one marker here. So if you add imaging, actually, you can get a lot more information and think about doing multiple intrinsic property measurements correlated on a per cell basis. So this is a toy example here, but we put in a bunch of different cells into a system, and then with imaging you can actually segregate them out and have maps of not only their electrical properties, but their size and their shape, and then ideally you can get better specificity for whatever it is the assay you're looking, the phenotype you're looking for. Okay, so I leave it back to here. Can these physical properties of tumors, and specifically the electrical properties, though we can think about other ones adding in, be useful in cancer? Thank you.
Questions? Jerry?
Explain a little bit more about how you multiplex this. How do you do flow and electrical measurements? Is it sequentially or?
That's a good question. Well, when you say flow, you mean flow cytometry.
Yeah.
Well, that's not what we're doing here. We're doing flow, period, meaning that they're in a little channel and they're flowing by and we're imaging. So we're doing essentially bright field microscopy while we're doing the measurement, okay? Now, could you then go in and do fluorescence at the same time? Probably. So it's not that we're taking this and putting it into a flow cytometer, we're doing that assay and putting it into microscopy.
Mike Beer, I think, had a question.
Have any idea how sensitive this is?
Sensitive in what respect?
Well, can you find a cell in a million?
That's a very good question. I don't know the answer to that, except I'll say that there is a company that is working on this for CTCs, and they just delivered not this, but they're working on using electrical properties to isolate CTCs, and they've just sent a product to the NCI.
Yeah, please.
Does this only work for cell surface proteins? Or is the cell kind of a Faraday cage?
Thank you for asking that question. In fact, by varying the frequency, you can probe the envelope, you can probe the cytoplasm, you can probe the nucleus, and so you can actually get some specificity in the compartment by altering that frequency. And we actually did a E-screen, both probing for different compartments.
If there are no other questions, let's thank Dr. Voldman enthusiastically.